When most people imagine evolution, they picture a family tree. Parents pass DNA to offspring, mutations appear over generations, and natural selection slowly shapes each branch. That vertical inheritance is real, and it explains much of life's history. But it is not the only way genes move. In many organisms, especially bacteria, DNA can travel sideways. A cell can acquire useful genetic material from another organism that is not its parent, sometimes not even a close relative. This process is called horizontal gene transfer, or lateral gene transfer. It turns evolution from a simple tree into something more like a web, where branches can reconnect, exchange tools, and change direction surprisingly fast. The key idea is simple but powerful. A gene is a biological instruction, and under the right conditions, that instruction can be copied into a new host. If the new gene helps the recipient survive, it may be preserved and passed on to later generations. This is very different from waiting for a helpful mutation to appear by chance inside one lineage. Horizontal gene transfer lets organisms borrow existing solutions. A bacterium facing a toxin, antibiotic, or new food source may not need to invent a defense from scratch. If another microbe nearby already carries a useful gene, evolution can sometimes move that gene across the gap. This is one reason horizontal gene transfer matters so much in bacterial evolution. Bacteria reproduce quickly, but they also live in crowded environments: soil, oceans, intestines, hospital surfaces, wastewater, and biofilms. In those places, many species exist side by side, constantly releasing DNA, exchanging mobile genetic elements, and interacting with viruses. That creates opportunities for genetic traffic. Instead of each bacterial lineage evolving in isolation, useful traits can spread through whole microbial communities. The result is a form of adaptation that can be much faster and more networked than the slow, strictly parent-to-child model people often imagine. There are several major routes for this sideways DNA movement. In transformation, bacteria take up loose DNA from their environment, often from dead cells that have broken apart. In transduction, viruses that infect bacteria, called bacteriophages, accidentally package bacterial genes and deliver them to another cell. In conjugation, one bacterium transfers DNA directly to another, often through a plasmid. These mechanisms differ, but they all produce the same evolutionary consequence: genetic material can cross from one organism into another. If that DNA becomes stable and useful, it can reshape the recipient's abilities. Plasmids are especially important in this story. A plasmid is a small, usually circular piece of DNA separate from the main bacterial chromosome. It can carry genes that help a bacterium survive stress, use unusual resources, or resist antibiotics. Some plasmids are mobile, meaning they can move between bacterial cells through conjugation. Because plasmids are modular, they can behave almost like biological toolkits. A bacterium does not just inherit its permanent genome, it may also acquire an accessory set of instructions that can be gained, lost, or passed to neighbors when conditions make those instructions valuable. Bacteriophages, the viruses that infect bacteria, add another layer. A phage normally uses a bacterial cell to reproduce itself, but during that process, pieces of bacterial DNA can be mistakenly carried along. When the phage infects another bacterium, it may introduce that borrowed DNA into the new host. Some phages are temperate, meaning they can integrate into a bacterial genome and remain there for a time. This can change the bacterium's traits, sometimes dramatically. In certain cases, genes connected to virulence, the ability to cause disease, are associated with phage activity. Viruses are not just predators in microbial ecosystems, they can also be couriers of genetic change. The most urgent real-world example is antibiotic resistance. If one bacterial strain evolves or acquires a gene that protects it against a drug, horizontal gene transfer can allow that resistance to move into other bacteria. This does not mean every transfer succeeds, or that resistance appears instantly everywhere. But it means resistance can spread across populations in ways that are difficult to control. In medicine, this is a major challenge. Antibiotics create strong selection pressure. Bacteria carrying resistance survive while susceptible ones die. If resistance genes are also mobile, treatment can unintentionally favor not only resistant bacteria, but the genetic systems that help resistance travel. This is why hospitals, farms, wastewater systems, and natural environments all matter in the antibiotic resistance problem. Bacteria do not respect the categories humans use. A resistance gene may move through environmental microbes, animal-associated bacteria, and human pathogens, depending on contact, selection pressure, and ecological conditions. Factors like antibiotic use, population density, sanitation, biofilms, and microbial diversity can influence which genes persist and which transfers matter. Horizontal gene transfer does not make resistance inevitable, but it makes the genetic landscape more connected. Managing resistance therefore requires thinking beyond one patient, one infection, or one bacterial species. Horizontal gene transfer is not only about danger. It also helps explain how bacteria adapt to new chemicals and environments. Some bacteria can degrade compounds that did not exist naturally in ancient ecosystems, including human-created pesticides or industrial chemicals. The ability to break down such molecules may depend on enzymes encoded by genes that can move, combine, and spread. In a polluted environment, microbes with the right genetic tools have an advantage. Over time, horizontal transfer can distribute those tools through the community. This is one reason microbial evolution is so important for understanding biodegradation, environmental recovery, and the long-term consequences of synthetic chemistry. For a long time, horizontal gene transfer was mainly associated with prokaryotes, especially bacteria and archaea. But evidence increasingly suggests that eukaryotes, organisms with complex cells such as animals, plants, fungi, and protists, can also be affected by gene transfer. These events appear less frequent and are harder to prove, but they can still matter. A transferred gene might help an organism exploit a new food source, tolerate stress, or interact differently with its environment. The challenge is that eukaryotic genomes are larger, more repetitive, and more complicated. Distinguishing a true transferred gene from contamination, ancient inheritance, or analytical error requires careful evidence. The existence of horizontal gene transfer changes how scientists think about the history of life. The tree metaphor is still useful, especially for tracking inherited lineages, but it is incomplete. In microbial evolution, life's history often looks more like a network, with genes moving between distant branches. This does not erase ancestry. Organisms still reproduce and inherit genomes. But it means that some traits have histories different from the organisms that carry them. A bacterium's ribosomal genes may tell one story about ancestry, while a resistance plasmid tells another story about recent exchange, selection, and ecological contact. There is also a deeper implication. Horizontal gene transfer may have helped maintain common biochemical features across life. All known life uses DNA or RNA, proteins built from amino acids, and a genetic code that is broadly shared. Early in evolution, before modern lineages were firmly separated, genetic exchange may have helped stabilize and spread core biological systems. Scientists still debate the details, but the concept is striking. Life may have become more universal partly because early organisms shared innovations. Instead of isolated experiments evolving separately, the earliest living systems may have formed a community where successful molecular solutions moved around. Horizontal gene transfer is evolution with shortcuts, but not magic. A transferred gene must enter a cell, avoid being destroyed, function in a new biological context, and provide enough benefit to be maintained. Many transfers fail or disappear. The important cases are the ones that survive selection and become part of a lineage's future. That is what makes the process so consequential. It explains the rapid spread of antibiotic resistance, the flexibility of microbial communities, the role of plasmids and viruses as genetic vehicles, and the web-like structure behind much of life's history. evolution is not only descent with modification. Sometimes, it is also survival by borrowing. If you found this helpful, please like this video and subscribe for more explainers.